Hello everyone and welcome to our module on T cells. T cells are part of the adaptive immune system, and there are millions of T cells in the human body, and each cell recognizes a unique antigen via its T cell receptor. T cells emerge from the thymus as naive T cells. They're called T cells because they mature in the thymus. B cells are called B cells because they mature in the bone marrow. Once a T cell encounters its unique antigen, they are called mature T cells. And a very important fact that you need to understand is that T cells recognize only peptides. So for example, the polysaccharides in many bacterial capsules are made up of sugars, and they cannot be recognized by T cells. Only peptide chains made up of amino acids and proteins can be recognized by T cells. T cells only recognize protein antigens when they are presented. So let's talk about antigen presentation. There are cells called antigen presenting cells, and they produce peptide fragments on their surface for T cells to react to. They place these peptide fragments on top of a molecule called the major histocompatibility complex. There are two MHC molecules, MHC type 1 and MHC type 2. We'll talk about that in this module. And T cells will only react to antigen when it's placed on self-MHC. This concept is called MHC restriction. So to understand this, imagine that we have some tissue here and there are little protein antigens floating around. Well, the T cell will not react to those at all, even though this T cell is uniquely designed to react to these protein antigens right here. What needs to happen is that another cell, called an antigen presenting cell, has to consume these protein antigens and then present it on its surface on top of an MHC molecule like this. Then the T cell can see it and react to it. T cells require this process of antigen presentation because it fine-tunes the immune system. If T cells could react to any antigen found in the tissues, they would probably overreact and we would die from autoimmune disease. This system of antigen presentation probably evolved to control the T cell response and make sure that it was only activated for the appropriate invaders of the body. Most T cell receptors contain two chains called the alpha and beta chain, and this is an illustration of that right here. And as you can see shown in this picture, the T cell receptor doesn't go very far into the cell. So in order to transmit the signal that the T cell receptor has bound its antigen, it needs some help. And that help comes from the CD3 complex, and that's all this material here that is surrounding the T cell receptor. This is a signaling complex, and it transmits the bound signal into the cell. And you can always think of CD3 as going together with the T cell receptor because it is responsible for transmitting the signal into the cell. So we have millions of T cells in our body capable of reacting to millions of unique antigens. How do we generate all that diversity? Well, the way it's done is by a similar process to antibody heavy chains, and I discussed this process in detail in the module on B cells. But just to summarize for our purposes here, there are gene segments called variable segments and diversity and joining and constant, and these rearrange in a random pattern to create the millions of diverse T cell receptors. There are also hypervariable domains of these genes that can change frequently, and again, this all adds up to create great diversity so that we have millions of T cells in our body capable of reacting to millions of potential antigens out in nature. There are two key subsets of T cells, and they're defined by the markers on their surface. One group has the CD4 marker on its surface, and another group has the CD8 marker on its surface. The CD4 T cells are called helper T cells, and their major role is to produce cytokines which will activate other cells, and they direct the immune response. Some people call them the quarterback of the immune response because they coordinate the reaction of the adaptive immune system. The other type of T cells are the CD8 T cells. These are often called cytotoxic T cells because their job is to kill virus-infected cells. They also kill tumor cells and are important for protecting us from tumors. So let's start by talking about CD4 T cells, and these T cells can be activated by antigen-presenting cells that possess MHC class II on their surface. We'll talk about MHC class II on the next slide, but it binds to the CD4 on the T cell. And the three types of cells that present antigen to CD4 T cells are dendritic cells and macrophages and B cells. And this is a little depiction of it right here. An antigen-presenting cell puts an antigen on top of an MHC class II molecule, presents it to a T cell, and then the T cell can become mature and can begin secreting cytokines to activate the adaptive immune response. MHC class II is a very important molecule. It binds to the T cell receptor and CD4, and it's expressed only on those antigen-presenting cells I just discussed. It's made up of two protein chains called an alpha and beta chain, and this is a picture of it here to help you remember. And it's very important that you understand the process by which MHC class II gets to the surface of antigen-presenting cells in order to present antigens to T cells. Imagine that we have a macrophage out in the tissues here. We'll just write macrophage here. And imagine that it has consumed a bacteria or virus and placed it inside of a phagosome. Well, that phagosome will merge with a lysosome and the bacteriovirus will be killed, and it will be broken up into little pieces, including some small peptide fragments that can eventually be presented to T cells for activation. MHC class II will be synthesized out in the cytosol, but a molecule will be placed on its surface called the invariant chain, and this will block anything inside the cell from getting into the binding groove of MHC class II. The class II molecule will then merge into the phagosome lysosome, and the invariant chain will be destroyed once it's inside, and then the MHC class II can bind to some of these protein antigens. It can then go to the surface, and you will get MHC class II on the surface of an antigen-presenting cell with a peptide fragment, and that is now capable of reacting with a T cell. So I've summarized these points here. It binds invariant chain in the endoplasmic reticulum inside the cytosol, and this prevents binding of intracellular proteins. It must then merge with an acidified lysosome where there are peptide fragments. The invariant chain will be released, and antigens can bind to MHC class II and then find their way to the surface. CD4 T cells require more than one stimulus in order to become fully active. This is a process called co-stimulation. There is a protein on the surface of antigen-presenting cells called B7, and it has to bind to CD28 on the CD4 T cells in order for the T cell to become fully active. I've illustrated that on the screen here. B7 and CD28 must bind in addition to MHC class II and the T cell receptor binding, and then CD4 interacting with both the T cell receptor and MHC class II. Both of these interactions must occur in order for the T cell to become fully active, and you will see this principle of co-stimulation commonly in immunology. This likely evolved in order to prevent T cells from overreacting and leading to autoimmune diseases. So what do CD4 T cells do once they're appropriately activated by their unique antigen and co-stimulation? Well, one of the things they do is stimulate B cells. B cells are much more effective antibody producers when they are co-stimulated by T cells. They can also trigger B cells to class switch and produce different types of antibodies. CD4 T cells can stimulate CD8 T cells, and they can also activate macrophages. It's very important that you understand that there are two subpopulations of CD4 positive T cells. They are called Th1 and Th2 T cells. They are both T helper cells, however they drive different immune responses from the immune system. The Th1 cells are the cell types responsible for what's known as cell-mediated immune responses. These are responses that consist predominantly of CD8 positive T cells and macrophages. In addition to the Th1 cells stimulating CD8 T cells and macrophages release IL-12, and this in turn drives Th1 production. And this type of T cell response, the Th1 response, is important for promoting certain types of specific IgG subclasses that function for opsonization and complement activation. Contrast that with the Th2 subpopulation of helper T cells. This subpopulation is important for what's known as humoral immunity. The Th2 cells activate B cells to produce antibodies, especially the IgE and IgA subclasses of antibodies, and thus they drive an antibody-mediated immune response. It's very important that you understand the different cytokines secreted by Th1 and Th2 cells and how those different cytokines drive the different immune responses. The two most important Th1 cytokines are IL-2 and interferon gamma. IL-2 comes mostly from Th1 cells, although some can come from Th2 cells. And IL-2 is a very powerful T cell growth factor. It stimulates the growth of CD4 cells and CD8 T cells. It also activates B cells and natural killer cells. And IL-2 is marketed as a drug called aldesleukin for renal cell carcinoma and melanoma. Some patients respond to this drug because it activates natural killer cells to kill the tumor cells inside their body. Interferon gamma is a very powerful activator of macrophages. It primes them to become better at phagocytosis and killing. It also activates Th1 cells and suppresses Th2 production so that it favors the Th1 response. And cells exposed to interferon gamma will express more MHC class I and more MHC class II. The important Th2 cytokines are IL-4 and IL-5 and IL-10. And of these, IL-4 is the major Th2 cytokine. Whenever you think of Th2 cells, you can think of IL-4, and that's easy to remember because 4 is 2 times 2. IL-4 activates Th2 cells and suppresses Th1 production. It also promotes IgE production, which is very important for the defense against parasites. It's also very important in the development of allergic diseases. IL-5 is important for activating eosinophils and helminth infections. It also promotes IgA production, and the IgA antibody is very important for mucosal immunity against GI bacteria. IL-10 inhibits Th1 production. It's very unusual in that it is an anti-inflammatory cytokine, and it has no pro-inflammatory effect. So the way to conceptualize the adaptive immune system and T cells is to think of a population of T cells, and the local cytokine environment is favoring formation of more or less of the Th1 or Th2 cell population. So things that favor the Th2 population I've written on the left side of the screen. IL-4 drives increases in Th2 cell populations, and IL-10 shuts down the production of Th1 cells. Both of these effects will favor a Th2 response. On this side of the screen, on the right side, I've written the things that favor a Th1 response. So macrophages will secrete IL-12, and that favors a Th1 response, and Th1 cells will secrete interferon gamma, which blocks the Th2 response. Another way to think of it is the way I've drawn on this slide, and you'll find diagrams like this in many textbooks and review books. So an antigen-presenting cell can present antigen to either CD8 cells or CD4 cells. The CD4 cells can release IL-2, which drives production of CD8 cells. CD4 cells can also release IL-4, which drives production of Th2 cells, which then go on to stimulate B cells and produce a humoral response. On the other hand, Th1 cells can get produced, which secrete interferon gamma, which activate macrophages, and in turn the macrophages secrete IL-12 to activate the Th1 cells. There's a little battle going on here between the Th2 cells and the Th1 cells. The Th1 cells are secreting interferon gamma, which shuts down Th2 cells, and in turn the Th2 cells are secreting IL-10 and IL-4, which shuts down the Th1 cells. For your boards, they're not going to ask you basic questions about Th1 and Th2 biology. They're going to want you to know how the Th1 versus Th2 response varies by the type of infection. And you should know that the Th1 response is very important for many intracellular infections. Intracellular organisms are protected from antibodies because they hide inside of other cells. So the Th2 antibody response is not going to be very useful. The Th1 cell-mediated response is going to be very important for intracellular infections. And one of the most important intracellular infections that is controlled by a Th1 response is Mycobacterium tuberculosis. Mycobacterium tuberculosis hides inside of macrophages, and the antibodies can't get in there. So you need a strong Th1 cell-mediated response in order to contain this infection. It's also very important to have a strong cell-mediated immune response for Listeria. Listeria is a facultative intracellular organism, and there are relatively weak Th1 responses in certain populations, and these are the populations that are at risk for Listeria. So newborns and the elderly have relatively weak cell-mediated immune response. They have weak Th1 responses, and these are the two groups who sometimes get Listeria meningitis. In addition, pregnancy is associated with a mild decrease in the cell-mediated immune system. This is probably to protect the fetus from immune cells. So pregnant women are at great risk for Listeria, and they can potentially pass it on to the fetus, and this is a condition called granulomatosis infantiseptica. In many granulomatous diseases, Th1 cells play an important role. There are many subtypes of granulomas. It's a complex topic. I've put a few references here on the screen if you want to read more detail. But in the prototypical granuloma caused in tuberculosis, Th1 cells play a major role. In general, granulomas are formed by macrophages, and as you may know, macrophages develop into giant cells, which are the hallmark of a granuloma. In the classic granuloma formed in tuberculosis, Th1 cells secrete interferon gamma to activate macrophages, and this contributes to the development of the granuloma. In addition, macrophages can secrete TNF-alpha, which is a very important cytokine that's been shown to promote the formation of granulomatous inflammation. Leprosy is an infection with the Mycobacterium leprae, and there are two forms of leprosy based on whether or not you have a Th1 or Th2 response. Tuberculoid leprosy is the milder form of the disease. These patients have limited skin lesions where they lose sensation over a patch of skin. These patients have a strong cell-mediated Th1 response that contains the infection, and if you biopsy the lesions, you see granulomas and relatively few mycobacteria. The other form of leprosy that is more severe is called lepromatous leprosy. This is where patients have diffuse skin lesions. This is a picture of a man with lepromatous leprosy on the screen here, and this condition occurs when patients, for unclear reasons, develop a Th2 response, a humoral immunity response, and they have depressed cell-mediated immunity. The antibodies can't reach the intracellular bacteria, and as a result, they get a much more severe form of the disease. There are two major forms of inflammatory bowel disease, Crohn's disease and ulcerative colitis, and they have two different T-cell responses. In Crohn's disease, biopsies show non-caseating granulomas, and as I said before, granulomas usually come from a Th1-mediated response. Contrast that with ulcerative colitis, where biopsies show abscesses and ulcers with bleeding. There are no granulomas, and ulcerative colitis results from a Th2-mediated response. Let's focus in on the relationship between Th1 cells and macrophages in the cell-mediated immune response. Macrophages secrete IL-12, which can stimulate Th1 cells, and Th1 cells secrete interferon gamma, which can activate macrophages. Rarely, children are born with a defective receptor for IL-12 on the T cell or a defective receptor for interferon gamma on the macrophages, and these children are susceptible to some unique types of infection. Rarely, children are born with a defective IL-12 receptor on their T cells. This means that IL-12 cannot trigger differentiation of T cells into Th1 cells. It also means you lose activated Th1 cells, which would normally produce interferon gamma to activate macrophages. As a result, there's a weak Th1 cell-mediated response and low levels of interferon gamma. These children have increased susceptibility to a number of infections. Classically, they are susceptible to disseminated mycobacterial infections, but they are also susceptible to disseminated salmonella, and these children have been reported to have reactions after they're vaccinated with the BCG vaccine for tuberculosis. And you can treat this deficiency with interferon gamma, and that should make sense to you because interferon gamma does what the Th1 cells would normally do, and it activates macrophages to help fight these types of infections. Another rare disorder is for children to be missing the interferon gamma receptor on their macrophages. These children also get severe disseminated mycobacterial disease. They can also get salmonella infections and others. This occurs in infancy or early childhood, just like the IL-12 receptor deficiency. What's important here is that interferon gamma is not effective. They do not have the receptor for interferon gamma. So the way you treat these children is by giving them continuous antimycobacterial therapy, and often they are referred for stem cell transplants to try and restore the receptors in their immune cells. Now let's talk about the other group of T cells, the CD8 positive T cells. And these T cells share many similarities with CD4 cells. They also react to unique antigens. They also require antigen presentation. And they also have a T cell receptor which is associated with CD3 for signal transmission. What's different about the CD8 T cells is that they do not react to MHC class II. Instead, they react to MHC class I, which is a different molecule that we'll talk about in the next slide. And unlike MHC class II, which is only found on unique types of cells called antigen-presenting cells, MHC class I is found on all nucleated cells in the human body. One of the most important groups that does not have MHC class I are red blood cells, but most other cells do. This means that most cells in the human body are potential antigen presenters for CD8 T cells, and their main role is to detect and kill virus-infected cells. To understand why CD8 T cells are so important, I want you to imagine that we have a cell inside the body and that a virus particle has gotten inside the cell and it's beginning to replicate and produce viral proteins and viral nucleic acids. Well, the entire time that this virus is reproducing, it would be protected from the immune system if not for CD8 T cells. Antibodies could not reach it because they cannot get inside the cell. Macrophages could not reach it because macrophages cannot phagocytose things that are inside a cell. And the CD4 T cells could not react to it either because for CD4 T cells to react to these antigens, they have to first be consumed by an antigen-presenting cell, and the antigen-presenting cells cannot consume particles that are inside another cell. So all these elements of the immune system would be unable to react to the virus, and it would replicate unimpeded inside of the cell if not for CD8 T cells. What CD8 T cells do is they react to MHC class I, which is found inside of all cells in the body. So this cell right here, where the virus is replicating, has some MHC class I inside of it, and it will go to the surface and place some of those viral proteins on its surface, and the CD8 T cell will see this, and the CD8 T cell will then react and kill this cell. So CD8 cells are extremely important for protection against viruses. If we didn't have them, the viruses would replicate unimpeded until they ruptured the cell and spilled out, and at that point there would be so many particles we could die from overwhelming infection. So let's talk about MHC class I in a little more detail. It binds to the T cell receptor NCD8. It's got a little bit of a different structure from MHC class II. It's got one chain called a heavy chain, and then another molecule called beta-2 microglobulin. This is a piece right here. The heavy chain has 3 subunits, and the beta-2 microglobulin binds right here, and it's got an antigen binding groove right here where proteins can bind. I've illustrated CD8 T cell activation on the screen here. An antigen presenting cell will present antigen in the context of MHC class I, which will then bind to the T cell receptor. And remember that almost any cell in the body can be an antigen presenting cell for CD8 T cells. CD8 will interact with the T cell receptor and bind to MHC class I. And then in addition, IL-2 can be secreted from T helper cells, and this in combination with appropriate binding can lead to stimulation and activation of CD8 T cells. So once a CD8 T cell binds to a cell in the body which has MHC class I on its surface and a foreign antigen, it can then kill the infected cell. And let's talk about how the CD8 T cells do that. One of the ways they do that is by inserting molecules called perforins. Perforins can form channels in the cell membrane, and this can lead to cell death. Another thing they can do is insert granzymes. These are protease enzymes that degrade cell contents. These can activate apoptosis inside the cell by activating enzymes called caspases that lead to programmed cell death for the infected cell. They can also insert a substance called granulysin, which has been shown to lyse bacteria, and it also can induce apoptosis. And then finally, CD8 T cells can produce a very important structure on their surface called the Fas ligand. The Fas ligand binds to Fas, which is CD95, on the surface of cells in the human body. This activates caspases inside the cytosol, and this leads to cellular breakdown. And this causes apoptosis, which is cell death with no significant inflammation. Apoptosis is programmed cell death. And the Fas ligand is an example of the extrinsic pathway of apoptosis. There are two pathways by which apoptosis can occur, and the extrinsic pathway is one of them, and CD8 cells induce this via the Fas ligand on their surface. Another group of T cells you should be familiar with are called regulatory T cells. What their role is precisely in the immune response is not well understood. It is known that they can suppress CD4 and CD8 functions. That's why they're called regulatory. They all express a molecule called CD25 on their surface. This is the classical marker of regulatory T cells, and it's made up of pieces of the IL-2 receptor. All the regulatory T cells have CD4 and CD3 molecules on their surface, and they're known to produce anti-inflammatory cytokines. And remember, IL-10 is a very important anti-inflammatory cytokine, and another one is TGF-beta. Another group of T cells that we don't really understand yet are the Th17 cells. These are a subset of CD4 T cells that are distinct from the Th1 and Th2 subsets. They appear to be very important for mucosal immunity in the GI tract, and they produce IL-17. That's why they're called Th17 cells. They recruit neutrophils and macrophages, and when you lose these Th17 cells, GI bacteria often get into the bloodstream. That's why it's thought that they're important for protecting the GI tract from infection. E. coli and other gram-negatives have often been found in the bloodstream of animals that lack Th17 cells. There's also some emerging literature that says that these cells may be important for autoimmune diseases. T cells are part of the adaptive immune system, and the adaptive immune system has memory, which means that a second exposure to the same pathogen elicits a quicker and more vigorous response. And the way this works in terms of T cells is that some T cells will become memory T cells. So you should know that most T cells involved in an immune reaction will die. The antigen will be withdrawn once the pathogen has been destroyed. There'll be a loss of stimulation. Cytokines like IL-2 will go away, and as a result, the T cells will undergo apoptosis. But a few will remain as memory T cells, and these cells can live for many years. And as a result of these memory T cells, a secondary response to a pathogen requires less antigen. In addition, a secondary exposure to the same pathogen produces more cytokines. And as a result, you get a faster and more vigorous response the second time you're infected with the same bug. One of the best examples of T cell memory is the PPD test. PPD stands for Purified Protein Derivative, and it's a test done to see if a person has been previously exposed to Mycobacterium tuberculosis. In the PPD test, you inject the tuberculin protein under the skin. If the patient has been previously exposed to tuberculosis, memory Th1 cells will be activated. They will secrete interferon gamma, which, as you know, is a Th1 cytokine. This will activate skin macrophages, which is what Th1 cells do through interferon gamma. And as a result, there will be local skin swelling and redness if the patient has a prior TB exposure. If the patient has no prior exposure, they will have no memory T cells, and as a result, there'll be no skin reaction. And this is an example of a delayed type hypersensitivity reaction, which I discuss in great detail in the module on hypersensitivity disease. You should be familiar with the concept of superantigens. These are rare antigens that can activate a massive number of T helper cells. So as you know, normally antigens are presented by antigen-presenting cells to a T cell, but a superantigen can directly link MHC with the T cell receptor and elicit a T cell response without being processed by an antigen-presenting cell. As a result of this, these superantigens can activate many, many more T cells than a normal antigen. Just to put this in perspective, a typical antigen elicits a response from less than 1% of all the T cells in the body. A superantigen can elicit a response from 2 to 20% of all the T cells in the human body. As a result, you get an enormous release of cytokines, especially interferon gamma and IL-2 from Th1 cells, and this results in massive vasodilation and shock. Patients exposed to superantigens can get the toxic shock syndrome, and this is a result of the massive stimulation of T cells. There are two bacterial toxins that cause the toxic shock syndrome. Staph aureus makes a toxin called toxic shock syndrome toxin, or TSST1, and Strep pyogenes makes a toxin called pyrogenic exotoxin A or C. Both of these can cause the toxic shock syndrome, and I talk about this in more detail in the infectious disease modules. For our purposes here, just know that these toxins result in massive stimulation of T helper cells. I'm going to finish this module by discussing the thymus. This is a structure found in the chest in the anterior mediastinum, and it is a site of T cell maturation. T cells come from the bone marrow as immature T cells, and they migrate to the thymus. Once they get to the thymus, they express the T cell receptor, and only those T cells with an ideal T cell receptor will survive. To be an ideal T cell receptor, you have to be able to bind to MHC class 1 and class 2, but you have to not bind in the presence of self-antigen, and most T cells that come to the thymus will undergo apoptosis because they don't have the ideal T cell receptor. This is a picture of the thymus and its location in the chest. Note that it's located in the anterior mediastinum of the chest. It has two lobes, and each lobe is surrounded by a capsule. And functionally, the most important parts of the thymus are the outer portion, called the cortex, and the inner portion, called the medulla. So as I said before, most T cells will die in the thymus. Those that are going to survive are selected for survival. So in the cortex, a process takes place of positive selection of T cells. Thymus epithelial cells will express MHC complexes, and the T cells are tested for binding to self-MHC complexes. Those that have weak binding undergo apoptosis. Those that have strong binding will survive. This is called positive selection because we are selecting for those cells with the strongest binding to MHC complexes. After cells survive the cortex, they migrate to the medulla, which is the inner portion of the thymus. In the medulla, a process called negative selection occurs. Thymus epithelial cells and dendritic cells will express self-antigens in the context of MHC. T cells are tested for their binding to self-antigens in MHC. If they bind too strongly, they will undergo apoptosis. This is a very important process to prevent autoimmune diseases. If we allowed T cells to survive the thymus that bound very strongly to self-antigens, we would get lots of autoimmune disease. And this is called negative selection because we are allowing cells to survive that do not bind very strongly to self-antigens and MHC complexes. So here's a picture to help you understand this process better and to help you remember it. T cells initially enter the subcapsular zone of the thymus, which is the outermost portion of the thymus. The T cells in this area are very immature. They do not express T cell receptors or CD3. They also do not express CD8 or CD4. The T cells then migrate down into the cortex. In the cortex, they begin to express T cell receptors and CD3. They also express both CD8 and CD4. This is the only place in the body where you will find T cells that express both CD8 and CD4 together. This is where positive selection takes place, so these T cells must bind very strongly to MHC 1 and 2. If they have weak binding, they will undergo apoptosis and they will die. Next, the T cells migrate to the medulla. In the medulla, they differentiate to be either CD8 T cells or CD4 T cells, and they're tested for binding to MHC and self-antigens. If they bind very strongly, they will die, and this is the process of negative selection. I'll finish this module by discussing the AIR genes. AIR stands for autoimmune regulator, and the AIR genes are responsible for the expression of self-antigens by epithelial cells and dendritic cells in the medulla of the thymus. So if you have a mutation in an AIR gene, this can lead to autoimmune disease due to poor expression of self-antigens, and there's a lot of scientific literature of people trying to understand how AIR genes work and how they might contribute to the development of autoimmune disease. The clinical consequences among people we know have AIR gene mutations are very strange and something that you just have to remember. Patients with AIR gene mutations develop recurrent candida infections. They get a condition called chronic mucocutaneous candidiasis. I talk more about this in the module on immunodeficiency syndromes. In addition, the autoimmune consequences of AIR gene mutations, for reasons that are poorly understood, appear to be directed at endocrine glands. So patients with AIR gene mutations develop hypoparathyroidism and adrenal insufficiency. And that concludes our module on T cells.